Imagine waking up and dreading the stairs in your own home. For millions of people with chronic knee pain, even getting out of a chair requires a grimace and a deep breath. If you look at this demographic breakdown of the global population, the scale of the problem becomes clear. Right now, osteoarthritis affects roughly 1 in 6 adults over the age of 30. A healthy joint relies on cartilage. It functions as nature's shock absorber, a smooth, almost frictionless cap that lets our bones glide past each other seamlessly. But this cross-section reveals the biological flaw that makes arthritis permanent. On the left, human skin is packed with blood vessels ready to heal a scrape. On the right, joint cartilage is a microscopic dead zone. It has almost zero blood supply. Because it lacks that vascular plumbing, cartilage heals incredibly poorly. Once standard wear and tear degrades the smooth surface, patients are trapped on a one-way street toward bone-on-bone deterioration. Our current medical responses focus on masking the symptoms. A doctor might prescribe painkillers, administer steroid injections, or lubricating gels. These can bring a few months of relief, but they do nothing to rebuild the tissue that wore away. When the joint eventually grinds down to total failure, patients face the operating room. Millions of total joint replacements are performed worldwide every single year. To understand this, look at this frontal X-ray. The surgeon cuts away the biological joint, hammering heavy metal hardware over the femur and tibia. From the side profile, the mechanical invasion is obvious. A massive metal stem is driven deep into the shin bone. We think of modern orthopedics as a highly advanced field, yet the core concept remains anchored in historical mechanical amputation. We are treating a complex biological failure with a brute force hardware solution. This approach suggests a need for a shift in how we view joint repair. Scientists in Colorado are currently leading a collaborative effort to move away from these mechanical replacements entirely. Their objective is to deliver a single injection that directs the human body to repair its own damaged cartilage from scratch. The first experimental therapy takes an already FDA-approved drug and alters how it enters the body. They package the medicine inside tiny biodegradable particles and inject them straight into the knee. Look at this release timeline. Instead of flooding the joint all at once, these microscopic particles deploy the drug in steady, therapeutic bursts over several months. It is similar to setting up a drip irrigation system to slowly water a plant, rather than dumping a bucket on it instantly. This sustained delivery solves a massive roadblock in orthopedic medicine. It stops the joint from rapidly flushing the medicine away before the tissue has enough time to trigger the healing process. The second therapy functions as a complete biological repair kit, utilizing a specialized gel loaded with engineered proteins. Doctors inject this liquid directly into the pockets of damaged cartilage. Once it settles into the defect, the gel rapidly hardens to form a structural scaffold. The material acts as a homing beacon. It recruits immature progenitor cells, specialized repair units that are already wandering naturally through the surrounding tissue, pulling them straight into the scaffolding. Once those cells arrive, the biomaterial issues a sequence of chemical commands. It instructs them to build brand new, healthy cartilage exactly where the bone was wearing away. This procedure effectively reassigns the task of repair to the body's internal construction crew, using biology rather than hardware to close the defect. Animal studies provide the initial evidence for this mechanism. This side-by-side comparison illustrates how a single injection regenerated a thick layer of cartilage and healthy underlying bone in just 4 to 8 weeks. The researchers also verified that this regenerative mechanism successfully commands human cells, extracted from patients who recently underwent joint replacement surgeries. However, a distinction is necessary. These results occurred in isolated human cells in a laboratory environment, not inside a living patient walking on a damaged knee. Before this becomes a standard treatment, researchers must conduct carefully controlled clinical trials. They need to find the ideal dose, prove the safety profile, and confirm if this newly grown cartilage will last for years. While a cure is not available today, successfully proving the biological mechanism in the lab is often the most difficult hurdle in biomedical science. The momentum backing this research is significant. The project recently secured a $33.5 million grant from ARPA-H, and a startup called Regeneration Therapeutics has been formed to move the therapy toward clinical use. If the upcoming safety data remains positive, human clinical trials could begin in approximately 18 months. The long-term goal is to provide a viable alternative to the operating table, where chronic pain might be managed with a single, minimally invasive injection. As these therapies advance, metallic artificial hips and knees may eventually be viewed as products of a more primitive period in medicine. Advancements in biomedical engineering are shifting focus away from stronger mechanical hardware and toward the re-engagement of the body's natural healing systems.